So welcome and thank you guys for attending our first SciComm live. Today we're meeting to discuss the implications of the recent controversial use of CRISPR-Cas9 to edit and subsequently implant human embryos. Today's discussion may be a little different than some of you are used to. Though this is a bona fide scientific discussion, it is also geared to the public, and from the videos and the audio around the room, it's currently being streamed to Facebook Live, and those on Facebook interacting can ask questions, which Sam will moderate, and I'll moderate questions from you folks. I first want to give a quick synopsis of the events that have led us here while we're discussing this. Then Erica will present a few slides on CRISPR, its mechanism, how it works, what it is, give some historical context, as well as its capabilities. And this will be generally geared for those, for the general public. And that'll be about 10 to 12 minutes. Then Samantha will introduce our panel members, and then we'll get into the discussion. So what led us here? CRISPR was first published on in the context of genome editing about 7 years ago. And since that time, it has really rapidly been taken up and utilized by the scientific community. And it seems like every week there are new CRISPR-mediated breakthroughs that are being published on. But the events that brought us here, these happened more recently. In fact, exactly 2 months ago, one of the inventors of CRISPR, Jennifer Doudna from UC Berkeley, the day after Thanksgiving, received an email from a researcher that was unknown to her and to the world, Dr. Hiv, and it just said baby's born, and that was it. And over the coming days, what emerged was the story of this researcher who essentially went rogue and used CRISPR-Cas9 to edit embryos, which goes against all stated scientific standards, ethical and clinical standards. He used these to modify. He used these to modify the embryos and subsequently implanted these using IVF, in vitro fertilization, which is a commonplace technique in reproductive assistance. Dr. Hiv knocked out a receptor, CCR5, which is believed to be the receptor that the virus HIV uses to gain access into T cells. Therefore, theoretically, he made these babies immune resistant to HIV. Around that time, he was summoned to Hong Kong at the Second International Summit on Genome Editing to explain his actions and to try to shed some light on what he did. And this resulted in the committees issuing a very strong statement against the researcher. His actions have been condemned swiftly by the scientific community. But nonetheless, now we're left with, what do we do now? How do we move forward? And so many prominent U.S. scientists have called for large-scale discussions on how to put policies in place and how to move forward in a way that prevents irresponsible genome editing in the future while still maximizing its benefits. And so that's where we are today. So I thank all of you for coming to participate in this. Now, Erica, not a PhD candidate from the Department of Biochemistry, she's going to present a few slides to kind of catch up the public on CRISPR before we get into our panel discussion. Thank you.
Okay, is the audio any better? since I have a microphone, if this is better, just let us know through Facebook Live if we still need to be working to fix the audio. Anyway, hi, I'm Erica. So I'm going to be giving a brief introduction into designer babies, not designer fashion babies, as the slide may indicate. But, forgot to turn my pointer on. There you go. Anyway, so I'm going to give an introduction into the science that was used to generate these genetically modified babies that have now been born, and where the technology comes from and how scientists are looking to use it and how it was just used. So the first thing we have to talk about is the basics of how DNA works structurally and functionally in the cells. So DNA is structured as a double helix, which means that it's made up of two strands that are twisted around each other. These strands are held together by the fact that they have different what's called bases on the inside, which you may have heard the terms A's, T's, C's, and G's, and that's just abbreviations for the names of these molecules that are on the inside of the DNA strand. So the A's pairs with T's and the C's pairs with G's, and that's like the different bases on the inside kind of molecularly hold hands with one another, and that keeps the DNA in one piece so that the different strands are wound around each other. And all these bases are lined up on a backbone that keeps the strand long and intact. So just remember this structure for later when we're talking about how we edit these bases or a strand of DNA as a whole. So the reason that scientists want to be able to change DNA is because changing DNA results in a change in RNA and ultimately a change in a protein, either that the protein ends up different or not being produced at all. So the way scientists make these changes is going with a new gene editing system called CRISPR. But in order to talk about that, we have to first understand a few things about bacteria. So, nope, that's not it. There you go. So most bacteria have a single circular piece of DNA as their genome. And when a bacteria gets infected by a virus, one of the first things that the virus can do is insert its own DNA and then chop up the bacteria's DNA so that the bacteria's protein-making machinery then focuses on making virus proteins. But just like humans have an immune system to try and combat viral infections, the bacteria kind of have their own immune system. They try to combat the viral attack with the technique that we're going to talk about that scientists have used and called CRISPR-Cas9. So what they want to do, the bacteria want to do, is they want to stop the viral DNA from being available as a template and also for the degradation of the host DNA and also for the generation of the machinery from the virus. So what CRISPR is, you'll hear that term a lot, but what it actually stands for is a region on the bacterial genome that contains a lot of clustered, regularly interspaced, short palindromic repeats. And the way you get these spaced repeats and why they're useful to the cell is that you get the bacterial genome, the virus genome, and on that is a region that we call a protospacer. And the reason that we call it a protospacer is because eventually it will be separated from the viral DNA and then incorporated into this region of the bacterial DNA. Now the bacterial DNA has a region, has a cluster of these regularly interspaced regions that are repeats, and they're palindromes, and that's just like the A's, C's, T's, and G's are set up as a palindrome. But the reason that you want to keep this is because now this region matches up with the virus that has tried to attack you. So you're keeping a list of what all the viruses that have made it into the cell. As I like to call it, bacteria's mafia-style hit list. Because of the way that bacteria reproduce, basically as clones of one another, the genes that get inserted into this CRISPR region of one bacteria will be carried on down to its descendants. So they can also recognize and protect themselves from these viruses. So you can have a region, the spacers in between these repeats, and the spacer from this was from a virus that had infected a generation above, or 2 generations above, or the bacteria itself. And the reason that this is useful is that you remember how I mentioned that the DNA gets transcribed into RNA. So just as part of the general process in a cell. When the bacteria as a whole detects the presence of foreign DNA, it makes strands of RNA from the DNA, kind of activates those genes or sequences, turns them into RNA, and then one of those pieces of RNA has a sequence that matches the invading DNA. So the RNA that you've produced, the bacteria has produced, will associate with this enzyme called Cas9, and the sequence that matches up with the viral DNA is called a guide RNA. And the guide RNA will form a complex with the viral DNA because it has that same sequence, because it was made from the invading viral DNA either earlier in that bacteria or in a previous generation for that bacteria. So it checks its hit list. It says, I know you. I've got a grudge against you. So it binds up kind of what I said earlier about molecularly holding hands, where this guide RNA, if the sequence is exact enough, it will cause the DNA to unwind as this strand of DNA kind of holds hands with the RNA instead of the other strand of DNA. And what this does is this guide RNA, by being complexed with the Cas9 protein, has guided the molecular scissors that this enzyme possesses to where it can cut the viral DNA. Now, when this cut happens, the cell's going to try to fix it. I guess in human cells, that would be to try and regain a function of the DNA. I suppose that the bacteria don't really care about fixing the viral DNA, but it just happens naturally. But the point is that the repair process is error-prone. And when you've got this double-stranded break, when it tries to fix it, it's not really great at it. The cell's not good. It's going to introduce some mutations and some errors, and that will lead to an activation of the viral DNA. So scientists use this in a very similar manner. They've found out that if you put in a strand of RNA that matches a target gene of interest, you can basically add a name to the Cas9's hit list. You can say, this is the gene we want you to target, and you have a guide RNA that will take the Cas9 that it's complexed with to your gene of interest and cause it to do a double-stranded cut on the backbone of the DNA. You can also introduce mutations by providing a repair template, but that wasn't done in the case that we're looking at right now, so you don't have to worry about it. And this technique is a lot easier for causing mutations than causing a mutation in the protein directly, because it's a lot easier to target a guide RNA that will bind your DNA of choice rather than trying to design proteins that are mutant proteins, because scientists really aren't good at designing 3D structures of proteins just yet. The issue about mutating an embryo is that when you mutate an embryo, every cell that comes from the embryo that only has mutant copies of the DNA, every cell that results from that is going to also have copies that are different than what the original programming read like. So you're going to end up with an entire organism with a change, and that person will also only be able to pass that change, that either deletion or that modification, to their offspring. So this is like a permanent change. So this isn't actually the first time that CRISPR has been used on human embryos. Back in 2015, Junju Huang used CRISPR on human embryos, but they weren't implanted. So he wasn't trying to generate actual living humans from this that would, have to deal with a lifetime of edits. And he was trying to fix a mutation that was causing a blood disease. So the difference is he didn't implant these embryos. And as Madison mentioned earlier, what Dr. Hay did was he actually used IVF to, the goal, ostensibly, was to create children who would be resistant to infection by HIV. And the way that he did this, kind of simply, as Madison mentioned earlier, was he deleted the gene for the protein CCR5. Now, CCR5 is believed to be a way in which HIV is able to access human cells and that by preventing the production of the CCR5 protein, you would block access of the HIV cell to the cells and make it where this person was immune to HIV, which sounds great as as the goal of your project, but there are some problems, and I'm sure that those will be discussed by our panelists. But very briefly, CCR5 may have roles, other immune functions, such as protecting against flaviviruses like West Nile, dengue, and yellow fever. There are also some suspicions that the change that was made can complicate symptoms of MS or other chronic conditions like hepatitis C and diabetes. And these are believed to be complications because sometimes this deletion does occur in nature. And people who have the deletion of the CCR5 gene often will experience complications from that. So I hope this is just kind of a brief introduction to how CRISPR works, and if you have any questions specifically for me through Facebook, just let me know. Otherwise, the more detailed information will come from our panelists, so I guess they're up.
Thank you, Erica. And again, thank you for those of you in the room tonight. And also thank you for everyone on Facebook tuning in. So for those of you who recently joined us, I'm Samantha Murphy, and I will be one of your moderators for tonight. I would like to introduce our panelists before we start our discussion, though. So Dr. Michelle Arnold is an assistant professor in the Department of Microbiology and Immunology at LSU Health Sciences Center in Shreveport, which is where we are right now. She is an expert in virology and understands how they can manipulate the immune responses. Dr. Todd Guidry is a postdoctoral scientist in the Department of Microbiology and Immunology as well, and he is an expert in virology and an excellent communicator of science. And Dr. Xiaohong Liu is an assistant professor in the Department of Pharmacology, Toxicology, and Neuroscience here. He is an expert in neuropharmacology who uses CRISPR for his models. So I would like to thank our panelists for volunteering for participating in this really neat event and welcome our live and online audience members. We now open our discussion.
So to kind of like start off, I'll ask, what is so controversial about exactly what was performed? Embryos have been edited in the past, and CRISPR has been widely used by many researchers. So what was different about this, and why was it so controversial? And anyone can take the question.
I'll be happy to start with that question. But I would like to give just a little bit of a preface by saying scientific researchers have spent a lot of time trying to build checks and balances into the systems of research that we do. So for instance, when we have results to publish, typically they go through a peer review system, meaning other experts in a related field, up to 3 or 4, will review your data, and then it's available for publication. But that means it's been looked at by other experts and deemed to be satisfactory in its content and the conclusions that it draws. That's on the tail end of our research, but on the front end of research, we also try to incorporate controls for the kind of experiments that people are doing. So for instance, if we are working in animal models, as Dr. Liu does, we have to fill out paperwork and get approval from a committee of experts who will judge the treatment of the animals and if the experiments are being done properly, so that no excessive harm comes to the animals and everything is being treated properly. The same goes for experiments that have humans involved. We want to make sure that people are not sort of going rogue, as we've implied in some of the advertising for this series, and you know, doing things without proper authorities' acceptance of it, and and without experts having reviewed it to say that yes, this seems reasonable, it's important, and it will yield useful results. So one of the controversies involved in this is that Dr. He, the scientist who has done this work, did not appear to seek the proper authorization for doing these experiments. So he has claimed that he received certain authorization from the hospital in which he worked to do these experiments. However, that hospital has denied those claims. We are not entirely sure of the status of that authorization, but that is one of the first concerns that has been raised about this work. A second concern is what is the outcome of these children who have been edited, right? What will their life be like? Will they be studied for the rest of their lives? The fact of the matter is we don't know very much. We don't know enough about the CCR5 gene alone to say remove it completely from humans and understand the outcomes of that. So we don't know now if these two new babies could have a lifetime of complications, or as some that were mentioned by Erica, we simply don't know the full extent of the side effects of deleting an entire gene. I don't know if you guys want to add to that.
Yeah, I think according to a Chinese news agency, he apparently forged ethical release forms to be able to get volunteers in his studies.
That's the current claim,
Yeah.
but there's no evidence on either side.
Yeah, right. Yeah, basically the biggest problem, or one of the main problems is that he did it without a lot of transparency. And, you know, like what Dr. Arnold said, we don't know what's going to happen to these girls. In Dr. He's video, he mentions that the babies were born healthy just like any other healthy babies, but we don't know what's going to happen to them as they grow up. And like what Erica brought up with the CCR5 gene is that there are people who do have mutations or deletions in the CCR5 gene that are walking around perfectly healthy, but as Erica brought up, there are some other complications. People with deletions in this gene do have an increased risk of very symptomatic West Nile virus infections, for example. And there are even like studies in mice show that if you take out this gene, the mice are more susceptible to brain injury during strokes, and there are a number of other studies showing complications with other issues. But even going further, it was actually determined that he edited, so whenever he edited the embryo, I think that was composed of several cells, and you actually don't see the same edit in the gene in every cell. Like some cells have a much bigger deletion in CCR5 than other cells, and we don't know what's going to happen or what are the consequences of those changes. Are they going to be different from a someone who has this naturally occurring CCR5 mutation?
Can I raise one
Yes.
more concern? Or do you want to? Okay.
I want to follow. Yeah, I think according to the recent, actually latest Chinese government issued investigation and their announcements, I think believe the nature of his conduct is a very blatant violation of national and international decree and laws. So actually, Chinese government harshly condemned such kind of research activity and issued a global ban on the human genome modifications activities. And I think even he himself is facing a criminal investigation right now. So I think what he did, all these scenarios sounds almost like a Wild West to me. Yeah, he forged the ethics documents and just to recruit those volunteer parents to attend the research, and he carried out all these experiments without knowing by the university. So all for his own fame and all for his own fortune. So this kind of conduct is definitely we need to harshly condemn. I think what he did here right now is modified the fertilized egg. The danger here is those genetic mutation will pass down to the future. If you make the future generations to the babies, if you make a mistake, that mistake will pass to all the future generations to change our human genome pool. There are two major worries here. The first is the off-target effects. So besides CCR5, you could make other unwanted mutations in the other part of the genome. For example, you could change your uncle gene to cause cancer. The second, I think you mentioned, is actually the modification is incomplete. So what he did is actually modify the fertilized egg at the single cell stage for the in vitro fertilization. At the single cell stages, you inject the DNA, you modify, use the CRISPR-Cas9 systems, but before he did the in vitro fertilization, those single cells have to be developed into blastocysts, which is composed of 300 to 500 cells. It's impossible to sequence all of these cells. You don't know what is happening with those 500 cells. Individual of those cells could develop in the part of your cells, in the tissue, in the babies. So that's the risk. Yes?
So this is a question for you, Dr. Liu, from Ryan Adler online. He would like to know if there are any residual effects on CRISPR-altered DNA that can be detected. So if someone's been edited by CRISPR, can you detect those changes?
So for detecting changes, right now current method is detect the off-target effects. Yeah. Usually what we do is we generate the genetically modified animal models using the CRISPR-Cas9 systems. Yeah, usually we will detect for using the online algorithm and the software, we can detect, we can predict what's the most vulnerable genes we can possibly make mistakes. Then we sequence those genome locus and to find out what's going wrong with those genome. Of course, we can do it using the next generation sequencing to sequence the whole genome and to find out unwanted gene. For animal models, this we can easily dilute out this unwanted genetic modifications very easily. We can breed out those mutants, only leave those mutants we want. But for humans, unfortunately, even you can, for the babies when they are born, even if you can detect those genetic mutations, those changes are permanent and will pass into the many generations and make a permanent change of the human genome pool. That's the risk here.
So when CCR5 is deleted naturally, as Erica mentioned, I'm assuming based on this discussion that it's deleted not from both copies of your CCR5. You still have probably some functional CCR5. Is that correct? Or whomever.
Yes. Mm-hmm. So actually, what they have detected from the cord blood from these two twin babies, they actually detect different kind of genetic modifications. What happens in one of the babies, I think, has 15 letters deletions in the CCR5 genes. In another baby, it has 4 nucleotides deletion. So for those 15 nucleotides deletions, it is a failed experiment because the whole gene is still in the translational frame, so that CCR5 still can be expressed. But for that nucleotide 4 deletions, you have created out-frame shift mutation, and therefore the whole genes cannot express, so you can inactivate the gene. But the problem is, what they are really trying to do, they're trying to mimic this delta 32 mutations in the CCR5 genes, which is spontaneous human in certain European populations that have this delta 32 mutations. What they generate, the 15 and the 4 nucleotides deletions, these are de novo mutations. nobody know what all these mutations were due. Okay, for example, that 15 nucleotides deletions, that whole gene expressed, but you don't know the function of the gene. It may increase the expression of the CCR5 gene, make the babies more vulnerable to the infection of the HIV. So basically, you have to provide the animal experiments to demonstrate that these two genetic mutations generated actually do not make any harm.
Can I add on to that? Because I think you were asking specifically about the natural mutations that are known to occur in humans in this gene. And as Dr. Liu mentioned, we know that that's a deletion in 30 amino acids, which is sort of known and somewhat understood that most of the protein product is still present and expressed in the cells of those individuals with a small removal of some of that protein. And we understand a bit about how that works, but we do know that that makes them, as Dr. Guidry mentioned, a little bit more susceptible to certain inflammatory diseases or to West Nile. There's some evidence for increased susceptibility and problems associated with those infections. So we know a little bit about this, what happens in the case of people who have natural deletions. They also appear to be resistant to HIV infection. That deletion has been known to occur either on just one copy of your genetic material or on both copies. It can be passed down on both copies.
So we have a question online from Katie. She would like to know how extensive was the changes claimed by Dr. Hay, and was it a single nucleotide change or more extensive than that?
She just said it was four base pairs deleted.
So is that what
Four
Dr.
base
Hay?
pair changes,
It was 4 base
nowadays
pairs?
15 base pair changes.
For both copies of the gene or one?
Yeah, they actually detect different versions of the genetic mutation. It is hard to know. It could be caused by two different situations. One is the CRISPR-Cas9 only deleted one single string. Okay, that's the others. The cells are mosaic in the blastocyst stages. When they implant the blastocyst, it is a mosaic, so you can detect a different version. With right now, their publication, their results are not for the peer review, the publication, so we don't know for sure.
You also had questions here.
If we talk about the CRISPR-mediated modification of the gene and not just deleting the gene, we may get the desirable outcome in the designer baby or the designer human. But the risk of that gene, let's say that we are doing a CRISPR-based repression of the transcriptional activity of a particular gene, and the risk of that gene being transcriptionally active in the subsequent generation all will be there. All right?
So the CRISPR remains in the subsequent generations after the CRISPR mutation or deletion.
Yeah, not the deletion, but just a modification, like we're trying to repress the expression of the gene in the designer baby, but that gene might be transcriptionally active in the subsequent generation. Because there are epigenetic changes in that.
It may be reactivated in subsequent generations.
But this is a deletion from chromosomal material, your actual inherited material, not specifically just to the RNA or the transcript that you're talking about, right? So your chromosomes are transferred from generation to generation, then the mutations will also transfer from generation to generation, unless they're otherwise edited again.
So are you of the opinion that the technique holds promise, but we first need to standardize these things in animal models and then maybe develop protocols to do humanized studies? But how much time will it take to develop something like this?
Yeah, let me answer this question. I think what's at risk here? Yeah, very important, we have to understand there are two kinds of genetic modification using the CRISPR-Cas9 systems. What he did is actually modified fertilized eggs, the germline modification. But for a long time, scientists actually have been using CRISPR-Cas9 in the adult cells. We call the somatic cells in cell body to modifications actually to treat human disorders. Like what we are doing in our lab is actually trying to develop the tools to modify the somatic cells. So for example, the tissue, the muscle cells, for example, the retina. So we make the changes only to this part of the tissue. The genetic changes will not pass to the babies, to the future generations. This is called the somatic cell gene editing. Sangoma, a company in Richmond, California, has long been avoiding target CCR5 genes using the somatic cell gene targeting. So what they did is they extract T cells, actually, in this case, from the 12 HIV positive patients, and take those cells out of the body and edit, get rid of the CCR5 gene, then put those cells, transplant those cells back. So their germ cells will not change. It will not cause mutations in the offspring. So this is somatic. So the lessons here, we are trying not to, pour the baby out together with the bath water. So we don't make the confuse this somatic cell gene editing with the transgenic, with the germline cell modification. So we will not lose our momentum to start the CRISPR-Cas9, use the CRISPR-Cas9 for the benefits of the mankind.
That's a much less controversial way of editing because, as you said, you're only editing one cell type, putting it back into, an adult patient, not an embryo. And so because only one cell type is being edited, it's not going to be passed on to future generations, so any mistakes aren't going to be passed on. And there's a fear among scientists who do that kind of work that this controversy is going to shed a negative light on that type of editing, of genetic editing overall, and set their field back.
Can you folks talk a bit about some of the bona fide uses of CRISPR-Cas9? It might be good to hear some of the uses that are being plans for its use, or I've heard about how this is a bad thing, but are there aspects of it or uses that are going to be positive in more detail? I've heard about agricultural uses for CRISPR-Cas9. Can you reiterate on any of those?
I think most importantly, I think it is very promises a very bright future for treatment of this a lot of devastating, currently incurable human disorders. What I think most important right now, we need to have some policy to oversee all this different CRISPR-Cas9 technology, so we can actually get rid of this work. This all current scenarios reminds me actually the similar actually scenario in 1970s. So in 1970s, with the rise actually the recombinant DNA technology, people actually reacted with the similar kind of worry, similar kind of panic. This actually directly led to the 1975 Arsenoma meeting organized by David Baltimore. So they published their recommendations, and those recommendations became the cornerstone used by the NIH and the FDA for their policy of the human DNA recombinant technologies, which guided for our research for probably the last 3 decades. I think we need to have similar kind of policy in place to guide our research, actually to educate the researcher and the trainees, to make them to understand, to clear about the risks and why such critical, the ethic and safety concerns are so critical. And I think most important, we need some kind of enforcement mechanism.
Can I just sort of address the question of sort of beneficial uses of CRISPR-Cas, right? It's not all about fear here. So in the case of, say, humans, we're talking extensively about human uses. One potential use would be to help assist with currently incurable genetically inherited diseases. For instance, Tay-Sachs disease, which is inherited from the parents to a child, and the child typically doesn't survive beyond a few years of age. It's possible that this would be highly beneficial down the road to saving children who have known genetically inherited diseases. Similarly, there are non-human uses that offer great benefit as well. For instance, if we consider organisms like mosquitoes or rats that are traditionally considered to be pests or maybe problems in agricultural industries, we may be able to modify these organisms and release them in order to reduce the populations of pests that might also be responsible for transmitting diseases. So these are some of the possibly beneficial uses of CRISPR-Cas that people are considering at the time. Although at the moment I can't say that I know of anything that is in extensive use because this is such a new technology.
There is a Editas, one of the companies, they have clinical trials for a type of genetic blindness.
Retinal disorder caused by genetic mutations. That is actually recently got approved. Last December, Editas got the first approval for the clinical trial of this therapy, and hopefully will be the first therapy actually using CRISPR-Cas9 technology to genetically modify the RCA10 mutations, actually, I think, only in the retinal. So it is a typical example of somatic gene editing.
So
Yes, okay, so Ryan would like to know, does methylation impact the efficacy of the CRISPR-Cas9 process?
I'm not quite sure about the specific methylation of what to actually cause the mutations, but even CRISPR-Cas9 right now has been developed systems actually to modify the methylation, importantly enzymes to modify the methylation by itself.
yes, sir.
So I'm just curious, if somebody in the US were to do what Dr. Haig did in China, are there possibly guidelines? Do we have laws? Would you be arrested? Would you be broken some law that would end up being in prison?
I don't think there is a law. There is a consensus about we cannot do this. Actually, 2 years ago, I think it's in Baltimore, there's the first international human genome editing meeting. In that meeting, there is a consensus, there's a moratorium, it's a temporal ban. So you can do the researches with the human embryos, but it cannot be used for the reproduction purposes. So this is the moratorium announced in that meeting. Okay, we do not have a current law, but I just mentioned it. I think head of the FDA, Francis Collins, and also the NIH, as well as the commissioner from the FDA, they all expressed their interest to have such kind of discussion, maybe to establish such kind of rule or law to govern this kind of research. But I would predict in the future, I think most of the research currently actually believe this is unavoidable. In the future, people will have to do the CRISPR-Cas9 in the human embryos to treat those incurable disease, even in the United States.
But would you,
Go ahead, I'm sorry.
Yes.
you would say there's not a law in which somebody
No.
would be arrested, but depending on their funding sources, right, there are potential negative outcomes
Negative
in the
consequence,
United States
absolutely.
because much research is funded by the US government. Again, there are rules in place, and if you were to break those rules, you risk the loss of your funding, correct?
Yeah, I know in China, one of the issues with wondering what's going to happen with Dr. He next is the fact that there's been a rule in China against this, but it's not an actual law, and there's no really guidelines on how to punish somebody for that. You have a question?
Forgery
Yes.
and that would be where they would get
Yes, the
in trouble.
forgery of the ethics documents, I think he's under criminal investigations for.
That is still under investigation though, and has not yet been proven that it's been fraudulent.
Yeah.
United
Could be.
States, that would be here.
Yes, ma'am.
So I know we've been talking a bit about the ethics of deleting CCR5. Can you guys discuss a little bit of the ethics of his experiments where he infected these children with HIV and the way he did it and then those long-term consequences?
That's a good question. Let's put it this way. There are 20,000 genes in human genome. That's right. 1/3 of these genes are actually brain-specific genes. As a neuroscientist, I know we know very little about all these genes. Okay? But all these genes are very important to control our learning, memory, emotion, personality, all the way to the consciousness. So you see what is at risk here is the humanity itself. You do not want to mess up those human genome to make the permanent genome changes to transmit those gene changes to the human genome pool. Because a lot of traits we have no idea about what these genes are capable to doing. That's the biggest risk of his experiments.
I'm not aware that he has tested the deletion. I don't believe that he has infected the children, the 2 children that were born with HIV. There's no ethical way of testing that. We don't know who the children are. They remain anonymous at this time. So we can't actually even go back to take samples from them to test to see what their genes look like at this time. So at this point, as far as I'm aware, and please correct me if I'm wrong, he has not infected them with HIV to test the knockouts.
Yeah.
Katie would like to know, to the panel's knowledge, has CRISPR-Cas been used in developing genetically modified algae for biofuels?
I don't know. I'm not sure exact case, but it's definitely possible.
Yes, it is.
There was a recent publication, like last week, actually. I didn't read it, I just.
There's another question. Is it known what kind of immune response is evoked due to gene modification in CRISPR babies? Like what is, with the loss of CCR5?
So let me say a little bit about what CCR5. CCR stands for chemokine receptor, meaning that this is encoded in the human genome and it plays a role in the inflammatory response. So this is a receptor expressed on certain cell types, meaning it's on the surface of the cells, and it recognizes signaling molecules that are sent out by other cells to attract immune cells to a certain area where maybe an infection or an inflammatory process is going on. We know that there are humans with small deletions, as I mentioned earlier, in this protein that's expressed on the cell surface. We don't really know the outcome of completely eliminating it from expression in these cell types. We really don't understand. But it does have, this protein has a normal role in human beings. It doesn't seem to be essential for survival, but it may have an impact on response to other infections or other parts of your normal immune response. We just don't fully understand the implications of it because simply not enough studies have been done. And we may never 100% understand everything that a single protein does because there are so many possibilities. I don't know if you want to answer that. Okay.
Yes, sir. As you mentioned that there can be so many proteins and then there are so many possibilities, right? As a graduate student, I'm all in for scientific rigor, but the general public looks for the results. There are so many FDA-approved drugs of which we do not know the mechanism or we do not know the complete side effect, but they are still out there and we take it because they are effective for at least the disease that they are used. How do you encounter general public opinion about this? If I'm taking a CRISPR shot and if it cures my AIDS, it prevents HIV, how do you encounter that strong opinion?
So, I mean, I would argue that the FDA has strict regulatory processes that manufacturers must go through in order to get approval for drugs, right? There are extensive studies at the level of just cells in the lab through animal studies that test the impacts of drugs on the animals, all the way through various levels of human-involved studies. And so in this instance, what you have is we have some understanding of this process, CRISPR-Cas, and but somebody has essentially skipped all of the regulatory steps to jump right into creation of a genetically modified human being. And without the proper regulatory steps in the meantime, you skip a lot of the things that might have been otherwise found in, say, an animal model, right? And so that's where the discussion needs to take place. What are the regulatory mechanisms that need to be put into place for CRISPR-Cas? And we would learn and modify along the way, but something needs to initially be put in place. And I don't know if you want to talk a little bit more about how animal models help us to understand things that like drug treatments before we put them into humans.
Very stringent regulations about toxicology studies, bioavailabilities, and potentially possible effects, tetragenic effects in the animal models before we can actually put into the clinical trials. Yes.
I don't want to sound as a conspiracy theorist here, but how many of these regulations are subjected to lobbying and financial biases?
I'm not a member of the pharmaceutical company or the FDA, so I can't really answer that for you.
Even so many drugs are out there which are basically toxins.
I would argue that's highly speculative and that you are implying that process is flawed. But I don't think that we really have the evidence for that. So I think it's a little bit dangerous to have that discussion, suggesting that these mechanisms that are in place don't work.
Yes.
So tonight we talked about the ability for CRISPR-Cas9 to efficiently or delete genes. How capable is CRISPR-Cas9 to insert new genes?
To insert new genes. Okay, as you know, the basic mechanism of the CRISPR-Cas9 actually just introduced by Erica. So just think about in a simple way. Like a human skin get cut. Okay, our body can repair it. The left scar. The same thing with the CRISPR-Cas9. Our double-stranded DNA can be cut by Cas9 enzymes. Okay, but it will leave a scar. And the cells has the mechanism to repair that scar. And that scar is what make the permanent changes of our DNA. That's how the CRISPR work. There are two major mechanisms for repair the scar. The first, which is actually what used in the health studies, is the deletion. Okay, we use the so-called non-homologous indwelling mechanism, which is error-prone mechanism very easily to make mistakes. That's what he did. Yeah, some with the 5 deletions, some with the 15 deletions, some with only 4 deletions. But we also have another mechanism, which is related to your questions, homology-directed repair, which is more feasible, more safe and pathways to repair the DNA damage. And using that mechanism, right now researchers can insert large from the short and to very large fragment of the DNA. The difficulty of doing that, you have to design along homology arms, okay? And that mechanism is still depends on the homologous recombination steps. And we know in a lot of cells, this homologous recombination step is very inefficient. In the mammalian cells, homologous recombinations happens 1 out of 30,000 cells. That frequency is very low. But a lot of researchers have developed a very smart mechanism to increase that efficiency, to make the insertion of the fragment of the DNA into genome actually more easily to do. Our labs actually also developed several methods actually to help insertion of the fragment of the DNA into the genome. In those even very difficult to do the homologous recombination cells actually make it happen in the brain cells, for example.
Yes, ma'am.
My question is to Dr. Arnold. I just wanted to know, are there other chemokine receptor proteins in addition to CCR5, and are their roles redundant?
So are you referring specifically to HIV infection? Okay. So a little bit of background about that. As we saw in the introductory slides, HIV was the burglar trying to get inside the house. And so that's sort of analogous to the virus trying to get inside of a host cell. And there are several receptors that are involved in HIV entry. And they are found on what are known as cells that express a molecule known as CD4. And so CCR5 is considered to be a co-receptor. And so these are basically sticky proteins that are on the outside of a cell, and the virus is using them to recognize the cell and say, this is the one that I want to invade. And so most circulating HIV viruses currently use CCR5 as the co-receptor, but there is also a population of circulating HIV that uses a different chemokine receptor known as CXCR4 as a co-receptor. So in principle, by deleting CCR5 from a human being, it would make them resistant to infection by only those HIV viruses that use CCR5 to get inside the cell. But because there are other HIV virus types circulating that don't use CCR5, they use CCR4, they could still infect that individual because there's different co-receptors depending on the virus type.
And the viruses are smart enough to adapt or undergo antigenic shift or drift mutations and still be able to infect cells which have CCR5.
So
I think there's like some strains of HIV are particular for CCR5 and some for CXCR4, and then some that are kind of a mix of both, right?
yeah, that's right. That's right. So those viruses that use CCR5, if they can't get into a cell because there's no CCR5 present, they can't complete their life cycle. So they don't have the opportunity to adapt, right? And so it would be those viruses that can get into a cell that has either CXCR4 or CXCR5 that could potentially adapt. But if you stop the virus from even getting into the cell, it really can't mutate and make any changes, right? It has to get through that entry barrier.
Is CCR5 non-mouse viable?
I'm sorry.
CCR5 no mouse viable?
Yeah.
Okay.
Yeah. Yeah, you can knock out both copies and they're fine.
We have another online question from Katie Mariak, who asks, have other CRISPR-Cas9 or CRISPR-Cas systems been used to modify human cell cultures? She talks about Cas12a or CPF1 that's been shown to work with similar accuracy to Cas9, but uses only a single CRISPR RNA guide instead of the two. So I was wondering if y'all could speak about any examples of other Cas enzymes that you may have used and how they've been modified, either with different, negase functions, things like that, that people could use.
I think recently, for example, I published Cas12b actually significantly increased the specificity, also reduced the size of those Cas9 protein. So you can actually package the Cas9 and the whole CRISPR systems in the AAV for better and more efficient delivery.
People using alternate enzymes that aren't Cas9 as well.
Yes.
Could you, so you mentioned that HIV uses CCR4 as a co-receptor, is that right?
CXCR4?
As a co-receptor?
Right.
So is that like a subset of the HIV population that uses that? Or is that contemplated just in general with HIV? And then secondly, what is, how important is that receptor relative to CCR5? Would it make more sense to theoretically target it as opposed to CCR5?
I think what makes CCR5 such a good target for gene editing is the fact, one, as Dr. Arnold mentioned, I think most of the circulating HIV strains are more adapted to bind to CCR5 versus CXCR4. But then another reason is that CCR5 is more or less dispensable when compared to CXCR4. I don't think, I think in mice, knocking out CXCR4 is lethal or embryonic lethal. So I don't think it's as dispensable.
A comment about the gene knockout with gene knockdown. Do you think it's more ethical to use gene knockdown because it may not change your germline?
Yes, you mean for the purpose of human cell genome editing? Yes, there is potential possibilities that we can increase the specificity as well as increase the, for example, one of the worries is, the biggest worries actually is the off-target effects. You will have to overexpress the Cas9 in the embryos and for very long times, which will keep making the targeting effects. So right now people are generating newest approaches using so-called dead Cas9. Actually, instead of getting rid of the dead Cas9, basically they do not make the cuts, the double-stranded cuts, okay, but they can be fused with other gene expression enhancer or repressors, okay, to make for the therapeutic purpose.
Anything from you? So I'll ask one last question. Do each of you think that this will hamper research on genome editing in humans? And what can scientists do to ensure that doesn't happen? So if you want to start, Dr. Arnold.
So discussions are just starting to take place about what to do in this situation, and probably they should have started much earlier before this actually occurred. So the natural reaction, I think, is to overreact and issue a moratorium on research. And in general, that's just taking a wide cut to all types of research and stopping it from happening until some decisions can be made. And that's been done before, and oftentimes progress is very slow, and it hampers research for the time that can extend into years. So I don't think that a complete moratorium on all research involving embryonic mutations using CRISPR-Cas is really the best route to go, but that's my personal opinion. I think we can have a slightly more measured response, make sure that the moratorium comes from where the implantation stage occurs, where we are actually creating new babies. That allows research to progress in embryos, but to not allow for the production of new human beings without understanding what kind of effects can happen. In the meantime, then research can continue while the discussions go on about the best ways in which to sort of regulate this type of research.
Yeah, I think it really highlights the importance, I think, of science communication, not only among scientists within the scientific community and developing, certain policies on or standards on how these things are done, but also communicating with, our government, because they may react to controversies like this. And I agree with Dr. Arnold. I don't think that we should basically just stop doing this type of research. I mean, there are people who are already out using technologies like CRISPR-Cas9 to edit the germline, either embryos or also sperm or egg, to fight diseases like ALS or Alzheimer's disease. So I really hope that this doesn't set the field back too far. But again, the whole issue is that what Dr. He did was done without a lot of transparency and a lot of oversight, and there's too many questions right now about how do we regulate how this science is done. And so hopefully this will spark a lot more conversations in that regard.
Yes. Let me end up with a final story, actually. In the first International Human Genome meeting, there was actually a dramatic note. At the end of the meeting, one of the audience members came to the microphone, holding her tears, talked about her experience giving birth to a son with the anencephaly, a brain disorder, genetic brain disorder, suffered from epilepsy for 6 days and died. So she concluded, if you all have this knowledge and skills to eradicate all these disorders, then we can do it. We do have this medical need, a lot of incurable disorders. So it is unavoidable. Human embryo modification and the therapeutic gene will be in realization. So right now, although the organizers of the international meetings harshly condemned his works, but overall, the consensus is trying to promote a more transnational, better transnational way to perform the human embryos for therapeutic gene editing studies and pave that way for in the future, real using the human embryos for the therapeutic gene. But with all this, we have to execute extreme caution because it is the human genome pools we are dealing with.
All right, well, with that, I thank all of you for coming today, for tuning in. I thank our panelists, and certainly Samantha. She had her seminar yesterday, but she still found time to essentially organize this entire event. So thank all of you.